Hello, let's learn about how to link antimicrobial resistance and diagnostic stewardship. The discovery of penicillin in 1928 dramatically changed human and animal health and well-being. Antibiotics are life-saving in serious infections. Most achievements in medicine, organ transplant, cancer treatment, major surgery attributed to the use of antibiotics. Now we see the end of pre-antibiotic era. The golden era of antimicrobials was manifested by key actinomycete natural products, other bacterial natural products, fungal natural products, and synthetic antibiotics. It indicates that synthesis was inspired by natural product. And from 19 to during the last century, from 1900 to 2010, we have seen many antibiotics being discovered, starting from the natural products. The first report of antibiotics by actinomycetes was seen in 1920. While in the golden age, we have seen MRSA first detected and plasmid-borne resistance to ceramonides. Within golden age, we have had antibiotics like lymphomacytes, streptococcus, and others. While we had carbapenems and muciporins monobacteriums in the year 1980s. Here we had into 2000 century the two dipeptides, fluorometals, oxygen. So this is how from one side there was a discovery of newer antibiotics. Also, we were seeing parallelly the emergence of antimicrobial resistance and antifungal resistance. The rise and fall of antibiotics is evident from this chart. The number of deaths versus the number of antibiotics plotted. The number of deaths is increasing while the number of new antibiotics is decreasing. The resistant bacteria are on the rise. Antibiotics are on the fall. A failing market between 1960s, as you can see from the charts, to 2000, no major classes were introduced. This article can be referred into as Trends in Microbiology journal. Antimicrobial is the ability of a microorganisms like bacteria, virus, fungi, and other parasites to stop responding to an antimicrobial. As a result, standard response becomes ineffective, infection persists and may spread to others. The copious prescriptions worsen the resistance problem. What are the consequences of antimicrobial resistance? Post-antibiotic era is imminent and threatens to reverse medical progress. Simple infections like sore throat and infections are becoming incurable. Higher chances of treatment failure and death. Longer treatment, higher cost of healthcare. Uncontrollable epidemics are the consequences of antimicrobial resistance. The horror and the importance that comes from running out of antibiotics is evident now. The factors that are responsible for the development and spread of antimicrobial resistance. Antimicrobial use is a driver of antimicrobial resistance. Overuse, misuse, and irrational use by doctors, patients, and chemists. Non-compliance and self-medication by patients. Use in animal husbandry, aquaculture, and agriculture. Irrational use, non-availability or misuse of diagnostic support, and poor infection control practices. Let us see the paradox. What are the paradoxes which are influencing the antimicrobial? This is access versus excess. Sophisticated technology versus poor or no basic diagnostic faculty. Then access versus need. Then there is a rational prescription versus self-medication. Then brand versus substance and quality. So these are the paradoxes that we are facing. It is multisectoral. Department of Health and Family Welfare, Department of Public Health, Department of Rural Development, Department of Education, Department of Women and Child Development. They all are taking measures towards one health actions. It's a multi-stakeholder responsibility. Professionals from the healthcare, the society, the researchers, and private practitioners and political agencies needs to come together in this fight against antimicrobial resistance. Vertical as well as horizontal integration is required in this fight. Multidisciplinary team is required in this fight against antimicrobial resistance. Multisectoral, multidimensional, and multifaceted problem across biological, technical, economical, regulation, education, and behavior. Antimicrobial resistance is a complex issue from human, environment, as well as animal food. Environment is from pharmaceutical farms, hospital, households, and for wherein we have unused asbestos. From animal food, where there needs to be treatment and control, disease prevention, and growth promotion. AMR, can it be halted? No, it can only be contained. The main priority is to prevent infection. Containment is the next best option. Antimicrobial resistance is a natural phenomenon. Never forget, antibiotics are not the only management. Awareness and understanding is required. Communication on an information education, communication material, education training, the knowledge and evidence of surveillance of AMR, laboratories, then infection control, surveillance of health crisis infection, animal health, community and environment, then optimal use of antibiotics, the regulations needs to be there in place for the access of antibiotics, antimicrobial stewardship needs to be in place, both across in agriculture, animal husbandry, and in healthcare settings. Newer innovations and research and development is required for the newer medicines and diagnostics. Innovations needs to be put in place against this AMR. There needs to be international collaborations, national collaborations in this fight against antimicrobial resistance. How we can control antimicrobial resistance is because the integrated stewardship model. Wherein we have antimicrobial stewardship, infection control, and diagnosis stewardship. It requires a balancing act. Save the patient by giving broad spectrum empirical tests, or be conservative and save the future patients from resistant organisms. Diagnostics are the single biggest potential game changer in the fight against antimicrobial resistance. The time delay in starting appropriate antibiotic treatment and patient care is a matter of life or death for serious infections, such as sepsis, pneumonia, and meningitis. This has been complicated by the fact that number of patients with multi-drug resistant bacterial infections for whom there are only very limited therapeutic options is constantly rising worldwide. Microbiology laboratory therefore plays a crucial role in identifying the infection agent as well as the potential resistance to antibiotics in order to help clinicians prescribe the most appropriate treatment with the shortest time delay. The diagnostic tools helps to identify the patients who have bacterial infection who will require antibiotic treatment. Then identify the organism causing the infection in order to determine the appropriate patient management. Determine whether the pathogen resistant to one or more antibiotics as a critical step to help the clinician prescribe the most appropriate antibiotic as early as possible. This is how there is an interaction between diagnostic and antimicrobial stewardship. There is a clinical evaluation of the patient, and then the investigations are ordered, and on the basis of the investigation, the diagnosis and the treatment is initiated. While we see that diagnosis stewardship is right test, right place, right time, antimicrobial stewardship is right interpretation, right antimicrobial, right time. While diagnosis stewardship is test centric or microbiology centric, antimicrobial stewardship is antibiotic or pharmacy centric. So there is a relation of interaction between diagnostic stewardship and antimicrobial stewardship. You can refer to the article from my secretary in Journal of Clinical Microbiology for the same. The steps that are involved in the monitoring evaluation of diagnostic stewardship includes planning first to do a SWOT analysis, that is strength, weakness, opportunities, and challenges analysis, situation analysis, resource and needs assessment needs to be done. Then the inputs or the resources needs to be put in place like fundings, local guidelines and SOPs, trained staff, microbiology lab facilities, and communication protocols. Then the process needs to be initiated like mobilization and management of funds, development or adaptation of SOPs, development and implementation of training materials, implementation of training courses, international quality assurance, agreed means and frequency of communication. Then we have output or results in terms of sustainable financing or resources available on a regular basis, common understanding of the protocols, staff trained leading to compliance, increasing specimens submitted to the laboratory, good lab practices, patient treatment and surveillance actions are performed in a timely manner. So this needs to be monitored on a regular intervals in form of key performance indicators. Then we have outcome from these processes, which is evident in form of patient treatment guided by timely microbiological data, resulting in safer and more efficient patient care. Then accurate and representative AMR surveillance data to inform treatment guidelines and AMR control strategies. That's all from this session about the linkage of antimicrobial stewardship and antimicrobial resistance. Thank you.